Okay, this is the second part of your chapter 12 notes on reproduction in prokaryotic cells. I briefly introduced this in the last slide, so we're going to slide through this pretty quick. I could probably combine these two PowerPoints, but I have yet to do that. So still in chapter 12, there might be some of this information in chapter 13 too. So DNA in prokaryotic cell is a single strand in a loop. There's no mom and dad DNA. It's sometimes attached to the cell membrane. It replicates, unzips, copies the DNA, makes two cells. They should be clones, which means they should be exactly alike, unless mutations occur or other recombinations we'll talk about. They may contain one or more plasmids. I introduced that tiny loop of extra DNA that are able to move from one bacteria to another, and that allows for recombination. Recombination just means recombining of genes into different organisms. Binary fission, asexual reproduction of most prokaryotes involves just one parent. DNA replicates, cell pinches in two. It can happen as fast as about every 20 minutes. Why would it stop dividing so fast? It could run out of food or space. And then we talked about budding and creating a bud. There you go. And then exchanging genetic material, because prokaryotic cells can do this to some extent. First of all, they can use plasmids, that's called transference, and we will artificially do this later in the year. Transduction, they use a virus to carry bacterial gene from one cell to another, so the virus picks up the gene and then carries it from one to the other. And then conjugation, two bacterial cells will contact each other and form a conjugation tube and exchange genes down this tube. It's not really a reproduction, it's just a way to exchange DNA and to get more variation, because this is what's going to help them survive longer. So just pictures of each of those. Transformation, here's a gene that's been inserted into the plasmid, and then it's going to be taken up, and then it's going to make copies of that in the future. Transduction, bacteriophage, or some kind of virus that is going to introduce genes from one bacteria to the other, and then conjugation forming a tube here where it can transfer DNA. So this is a picture of transduction with the virus. There's going to be some genetic material in here, and they're going to put it into that cell. And then maybe there's some genetic material here, and it's going to pick it up and then transfer it. This is a conjugation tube right here, and they can exchange DNA along that tube. So there's really four ways that bacteria can get genetic variation. One, their DNA can mutate. Transformation, the plasmid, they can transfer DNA by plasmid. Transduction, this is where they're using viruses. And conjugation, where they have a tube connecting them. And that's it. Made you very excited that that was the fastest one yet.